Good day everyone, I am Carlo Sangalang and I will be your partner in today's discussion. So in today's discussion, I have here my objectives. Number 1 is to learn what is RNA processing and RNA editing. Number 2 is to know what is nucleic acid metabolism and the types of metabolism. And lastly, to distinguish the difference of DNA and RNA metabolism. So first in the slide is the definition of RNA. RNA stands for ribonucleic acid. It is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. RNA and DNA constitute the four major macromolecules which are the lipids, proteins, carbohydrates, and nucleic acid, and are essential for all forms of life. So now that we are done with the definition of RNA, let's now move on what is RNA processing. So RNA processing are primarily involved in converting the genetic information contained in a cell. So the RNA strands is processed so that its introns are removed and the exons are pushed together to make a continuous shorter strand. So I had mentioned a while ago the word introns. So introns are the non-coding sequence which are being removed when an RNA matures, while exons is a coding section of a gene that contains the information required to decode a protein synthesis. Ibig sabihin, yung introns, they are the one who are being removed kapag magbubuo na tayo ng protein synthesis, and exon naman yung natitira para makapagbuo tayo ng protein synthesis. So in RNA processing, we have three components. So two of them are the 5-copying and 3-polyadenylation. 5-copying, 3-polyadenylation are two components where messenger RNA are modified during transcription by the addition of a cap to the end and a polytail to the free end. So as you can observe in the picture, those two components are being placed on both sides of a sequence where the 5-cap is used as a recognition signal for ribosomes to bind to the messenger RNA. So the last component of RNA processing is RNA splicing. It is a process that removes the intervening non-coding sequences of genes, which is the introns, from pre-messenger RNA and joins the protein coding sequences, which is the exons, together in order to enable translation of messenger RNA into a protein. So for example, dun sa RNA splicing, halimbawa, observe kapag nagpapalipad ng rocket, kapag umaakyat na siya sa atmosphere, there are some parts that are being removed. So see that as the introns, and yung natira naman, those are the exons, kung saan yung exons na yun together in order to enable translation of messenger RNA into a protein. So yung exon na yun ay siya yung nagagawa siya yung kasama ng messenger RNA para makagawa ng protein. So next in the slide is the three types of RNA processing. So number 1, trimming one or both of the ends of the primary transcript to the mature RNA length. Number 2, removing internal RNA sequences by a process called RNA splicing. Yun nga yung pinaliwanag natin kanina. And then number 3, modifying RNA nucleotides either at the end of an RNA or within the body of the RNA. So, ngayon nga, natapos na tayo sa RNA processing, pupunta naman tayo sa RNA editing. So, it is a molecular process through which some cells make discrete changes to specific nucleotide sequence within an RNA molecule after it has been generated by RNA polymerase. So, ano yung tinatawag nating polymerase? Sila yung mga enzymes na nagdadala about the information of a particular polymer, especially yung DNA and RNA. RNA editing, as the word implies, there will be some changes o merong mga pagbabago dun sa sequence ng isang nucleotide within an RNA molecule. So, RNA editing may also include insertion, deletion, and substitution na madidiscuss nga natin mamaya. Substitution of nucleotide within the RNA molecule. So there are two categories of RNA editing. Number one is addition or deletion. As the word implies, addition, new nucleotides are inserted in the original sequence. So for example, imagine na nagtatype kayo ng message tapos nagkulang kayo ng isang letter dun sa word na tinatype nyo. You happen to add a letter to complete the word. So parang ganun din yung addition. So addition can be especially powerful because it can cause frame shift na madidiscuss natin mamaya. So normally, when RNA is read to make proteins, it is read in groups of 3 nucleotides called codons. So in addition, if you add a nucleotide in the middle of a sequence, everything downstream gets shifted over 1. So yun yung tinatawag natin na frame shift, kung saan it is a change in the reading frame for the rest of the protein. So as well as the deletion, it is the removal of a nucleotide. Parang kaparehas din siya ng addition, pero in contrast with addition, na nagkakos din ng frame shift. So the second category of RNA editing is what we call substitution. Substitution, one nucleotide is replaced with another. So for example, kapag nagtatype kayo tapos wrong spelling yung na-type nyo, nagsasubstitute kayo ng ibang letters para makorek yung spelling. Parang ganun din yung substitution. However, in order to create a substitution, instead of cutting out the existing nucleotides and replacing it with a new one common way, this is done through deamination. So deamination is a type of substitution because the adenine nucleotides loses a chemical group called an amine group. Now, we are moving with nucleic acid metabolism. But first, let us discuss what is nucleic acid and what is metabolism. So nucleic acid are substances present in living cells such as DNA and RNA whose molecules are consist of many nucleotides linked in a long chain. So para nga makagawa ng isang nucleic acid, kailangan ng maraming nucleotides upang makabuo nga tayo ng isang nucleic acid. Ang metabolism naman ay ang breakdown of food and its transformation to energy. As simple as that. So now, let us discuss it as a whole. So nucleic acid metabolism is a process by which nucleic acids are synthesized and degraded. It is concerned with the synthesis and breakdown of purine and pyrimidine nucleotides. Nucleotide synthesis is an anabolic mechanism, while destruction of nucleic acid is catabolic reaction, wherein anabolic reactions involve the joining of smaller molecules into large ones. So yung mga example nga niyan ay yung combination ng monosaccharides to form a polysaccharides. Anabolic siya kasi yung monosaccharides are smaller particles ng polysaccharides. Example din niya ay yung fatty acids to form triglycerides, amino acids to form proteins, at yung sinabi natin, para mabuo ang isang nucleic acid, kailangan natin ng nucleotides. So, on the other hand, catabolic reactions, in contrast with anabolic reactions, is a type of metabolic reaction that takes place within a cell. So, kabaliktaran naman siya ng anabolic, wherein larger molecules are separated to form smaller molecules, as in the case of respiration where glucose is broken down to form carbon dioxide and water. So, example nga nito ay glycolysis, citric acid cycle, yung breakdown of muscle protein and breakdown of fat. Now, we are moving with nucleic acid metabolism. But first, let us discuss what is nucleic acid and what is metabolism. So, nucleic acid are substances present in living cells such as DNA and RNA whose molecules are consist of many nucleotides linked in a long chain. So, para nga makagawa ng isang nucleic acid, kailangan ng maraming nucleotides upang makabuo nga tayo ng isang nucleic acid. Ang metabolism naman ay ang breakdown of food and its transformation to energy. As simple as that. So now, let us discuss it as a whole. So nucleic acid metabolism is a process by which nucleic acids are synthesized and degraded. It is concerned with the synthesis and breakdown of purine and pyrimidine nucleotides. Nucleotide synthesis is an anabolic mechanism, while destruction of nucleic acid is catabolic reaction, wherein anabolic reactions involve the joining of smaller molecules into large ones. So, yung mga example nga niyan ay yung combination ng monosaccharides to form a polysaccharides. Anabolic siya kasi yung monosaccharides are smaller particles ng polysaccharides. Example din niya ay yung fatty acids to form triglycerides, amino acids to form proteins, at yung sinabi natin, para mabuo ang isang nucleic acid, kailangan natin ng nucleotides. So, on the other hand, catabolic reactions, in contrast with anabolic reactions, is a type of metabolic reaction that takes place within a cell. So, kabaliktaran naman siya ng anabolic, wherein larger molecules are separated to form smaller molecules, as in the case of respiration where glucose is broken down to form carbon dioxide and water. So, example nga nito ay glycolysis, citric acid cycle, yung breakdown of muscle protein and breakdown of fat. So, next in the slide is RNA metabolism. It is a broad term that encompasses all aspects of the RNA life cycle, including transcription, splicing, translation, transport, and RNA processing. So, in RNA metabolism, we have three cycles. Number one is transcription. It is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. So, anong purpose nun? Ang purpose nun ay for a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide. So, the number 2 cycle is what you call the translation. It is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. So, under translation, we have three stages. First is the initiation, second, elongation, and third, termination. In other words, we can call them start, make longer, and stop. These terms are describe the growth of the amino acid chain or the polypeptide. The polypeptide falls in an active protein and perform its function in the cell. So, the last cycle is transport, the process by which RNA molecules are actively transported from one location to another. We will now move to DNA metabolism. It is the process by which cellular DNA acid is maintained and includes both DNA synthesis and degradation reaction involved in DNA replication and repair. Under the DNA metabolism, we have DNA replication, wherein it is the process of copying a double-stranded DNA molecule. Both strands serve as templates for the reproduction of the opposite strand. Transcription and replication both involve in making copies of the DNA in a cell. Transcription copies the DNA into RNA, while replication makes another copy of DNA. That's all for today. Thank you and keep safe.